Dear friends, welcome to Biology Made Easy. Today we'll discuss Michelson and Stahl's experiment to prove that DNA replication is semi-conservative. In 1952, when Watson and Crick proposed their model, they suggested that the two DNA strands are complementary to each other, and when one strand is destroyed, the other is automatically copied. So there must be some relation. That means one strand has some capacity to replicate another. But they didn't prove it. And at that time, scientists thought over it and they proposed different models like conservative model, dispersive model and semi-conservative model. According to this conservative model, the DNA is exactly copied. But how it is copied, nobody knows. Just as, suppose we are in the market and some artist will come. He will ask, please stand there, I will take your sketch. It is just like that. This is the original DNA and this is the sketch. But there is no scientific proof. Similarly, come to the second one, dispersive. In dispersive, the new one which is synthesized, here you see some part is old, some part is new. Old, new, they are present randomly. There is no scientific basis. Then ultimately, it was again found that the two strands which are present, they separated and one new strand was synthesized. This is known as semi-conservative mode of replication, which was expected by Watson and Crick, and experimentally it was proved by Matthew Michelson and Franklin Stahl in 1958. So how they conducted the experiment to prove that it is actually replicating in a semi-conservative method, that now we have to discuss. For this purpose, they took E. coli bacteria. E. coli. And in this E. coli, they put it in one medium where they put ammonium chloride, and here the ammonium is 15, means it is heavy. Again, in this medium, they put one bacteria, sorry, the bacteria is there, they put one bacteriophage. Now, this bacteriophage will enter into this E. coli and it will multiply. But where from it will get the nitrogen? Now, in the surrounding medium, there is a ammonium chloride having heavy nitrogen. So, when this bacteriophage will enter it and it will multiply, after several generations, automatically its DNA will be heavy. So, for several generations, they cultured this phase in the E. coli, and ultimately they isolated one phase in which both strands have heavy nitrogen. Then they conducted the experiments further. This is the heavy DNA molecule. Both strands are heavy. And now they put this DNA in a solution where there is ammonium chloride, but here the nitrogen is normal N14. And when it E. coli multiplied itself, E. coli usually multiplies in each 20 minutes. So when it multiplied, it was found that one DNA has one parent strand and one new strand. Means one has a heavy strand, the other is new. Similarly, in the other strand is same thing. So how they came to know that one is heavy and one is light? That for that purpose, they used cesium chloride. In cesium chloride solution, they put the DNA and they carried out buoyant density centrifugation. So according to the heavy or light, it will settle down. So when in the first case, when the DNA having both strands heavy were centrifuged in this flask, you see they are just in the bottom because both strands are heavy. But when they put it in the ammonium chloride for the first generation, the two DNA molecules are slightly above because these are hybrids. One strand is heavy, the other is light. Then again, these two strands were put again in the normal ammonium chloride, which you can call the second generation. And how many strands are old and how many new, that was again carried out by the density gradient centrifugation using cesium chloride, and it was found that half of them were hybrids and the other half were light. From the diagram you can see the first and last one red color and the other is blue color is there, while the middle one two, they are just light and light. Again, when it was put for the third generation and again in normal ammonium chloride, then again there are 8 DNA molecules, and out of the 8, only 2 are hybrids, while the remaining are only light. So when again centrifugation was done, only 2 were in the hybrid level, and the light molecules, the density, and there the density very less, and the number of strands increased. Again in the 4th generation, if this experiment will be carried out, again the light molecules will increase while the hybrid molecules will remain same. So from this experiment, they proved that the DNA is replicating in semi-conservative manner, means one old parent is there and one new is synthesized. Now, if you observe these figures, this is the first generation. In the first generation, there are 2 molecules of DNA. If question will be asked, how many DNA molecules or what is the percentage of DNA having heavy isotope, what will be the answer? Naturally 100%, because there are 2 DNA molecules and both have heavy isotope. Here some students confuse, it is 50. Why it will be 50? Because the DNA has heavy isotope. Only if your strands will be asked, then it will be 50. But when DNA is asked, it is 100%. So in the second generation, there are 4 DNA molecules. 2 are hybrids and 2 are light. So it is 50%. And the third molecule, it will be 25%. Fourth generation, it will be 12.5%. And if when you go for several generations, ultimately you will find that only 2 DNA molecules will have they will be hybrids, having one strand heavy, the other is light, and the rest are only light. So from this they concluded that the replication of DNA is semi-conservative. Now we will discuss some questions. Now the first question. Again, before going to the question, I can tell you that this experiment was conducted in case of prokaryotes, but the same experiment was again carried out in case of eukaryotes by Taylor, Wood and Hughes in case of Vicia faba, or that is commonly known as faba beans. That means both in prokaryotes and eukaryotes, the replication is semi-conservative. So now questions. Now the number 1 question. In the third generation, the heavy, hybrid and light strands are in ratio of A choice 0:1:1, B choice 0:1:3, C choice 0:1:7, D choice 0:1:14 in the second generation. Now you come to the second question. In the second question, in first generation, how many percentage of DNA will have heavy nitrogen? A choice 100%, B choice 50%, C choice 25%, D choice 12.5%. Now, one third question. In which eukaryote the semi-conservative replication of DNA was carried out? A choice Garden P, B choice Faba bean, C choice soybean, D choice you can tell Drosophila. So these are the three questions I have given. After going through the video, you will be able to answer all these questions. So thank you very much for watching it. Have a good day.